Hello, this is a PowerPoint that I put together, and I think this is for Monday. What day is it? So this would be Monday the 16th, and I'm going to try to walk you through it, and I would like for you to use paddles to answer some of the questions, and then you can probably share them with the sub as kind of some feedback as we go through it. So we'll try this, and hopefully it'll work well in this format. So this is a presentation about Tasmanian devils in Tasmania. So they live off the coast of Australia, and they're endemic only to that island. And in this case study, we're going to learn that they have this disease that's really affected their population. This is what they look like here. You can see this Tasmanian devil. It's kind of a smaller animal, and I'll try to play a video. I'll see if that works in a little while. If not, maybe you can find the video online. So here's some background information about these animals. They do live in Tasmania, and it is a large island. It's right off the coast of Australia. And they were extirpated from Australia. What that means is that they did originally live on Australia, and they are marsupials, and then they were extirpated, so they moved and kind of became isolated there in Tasmania. And what we find is that when populations become isolated, that can affect their genetics. They are the world's largest carnivorous marsupial, and these are some of the things they eat. They eat birds, snakes, insects, and carrion is dead animals. So they feed on some of those things, and they weigh about 10 to 20 pounds, and they do use a pouch for reproduction. And they also make ferocious cries when they're fighting or defending each other, and if I'm able to play the video, I'll show you that. And they're also pretty aggressive. So all of this is important information about these mammals, especially when we're studying their disease that we'll look at. So I'm going to pause here, and you can also pause the video, and I'd like you to write some of this information down in your notebook. We will also have your paddles out, but at this point I want you to write some of this down in your notebook so you can refer to this later, and we'll look at things like their behavior and how that can affect the disease. So I'll give you a moment to write that down. And if you need more time, just pause it. Oh, and I missed this point, so here's another point. They also exhibit intra-specific competition. Hopefully that goes away in a second. There we go, intra-specific competition. We learned this term way back in like chapter 14 at the beginning of the year. And this is competition within a species. So this means that they're competing with each other. Interspecific was the other term, the opposite, and that's competition between other species. So maybe between like a coyote and a wolf or something like that. That would be inter, but intraspecific is between themselves. So that's another important part when we're looking at this disease and how it spreads. All right, so now let's look at the disease. The disease was first noticed in 1996. It might have occurred before that, but Tasmanian authorities noticed it then, and they saw these ulcers on the animals' faces near their mouth. And they figured out eventually that they were cancerous. And they saw that they grew rapidly, and they eventually caused mortality. And a lot of that was because the animal was unable to eat after it got some of these tumors on its mouth. And I'll show you what that looks like in a second. This was called DFTD, which means Tasmanian devil facial disease. And it knocked out their population by 50%. And that means, so back in 96, they were projecting that they could go extinct in 25 to 30 years. We're getting kind of to that point. And actually, I think that 25 to 30 years was when the studies were published, which I have the dates in a minute here. I don't want to throw you off. Let me just look at these dates. So 2006 is probably where that 25 to 30 year projection is coming from. And this is causing a huge loss in biodiversity, and it's really rare for a cancer like this to annihilate a population. I am going to pause the video here. Whoops, hold on. Hopefully this works. And then I'll try to pull up a video for you. Okay, so I'm back and I have a couple videos queued up for you. This first one is showing you the spread of the disease. So I'll play that for a little while and you can see where it started. It'll explain some of it and hopefully you can hear the sound.
These distributions. So it started in the far northeast corner of Tasmania and has been moving south and west ever since.
So this is like the ripple of death.
The ripple of death, yeah.
You can see it going down the island.
You can see it moving down the island. Every year it moves 10, 20 kilometres.
Like, can you show me what it does? Do you have a picture or something? So this is a Tasmanian devil head?
This is a healthy devil.
Okay.
Now, the other half of this devil, it's got a small tumour. And you can just see the tumour, there. So that's a very, very early tumour. In one single devil, in fact, it was a female devil.
That'd be patient 0, right?
That would be patient 0.
I'm not sure why it keeps pausing, but let me queue up this other one now. And this is just showing them fighting. So I wanted to show you them what their behavior actually looked like and how they were fighting. See if they get into it again. Here we go. Fast forward a little. Maybe. Okay. So that kind of demonstrates some of their aggression, and you can see that they would really get into it and get pretty aggressive with each other. And let me go back to this slide and make sure we looked at all of this. So some of this stuff would be good to write down for information. We do see that it is a cancerous disease, but it's a weird one. And it has caused a huge loss in biodiversity. And this is really rare. Cancers usually don't behave like this. They usually don't annihilate whole populations at this rate. Okay, so this is what the tumors actually look like. So that's a pretty good example of that. And you can see how that would cause death, why some of these little animals wouldn't be able to survive that. They wouldn't be able to eat and function. And so now on your paddles, I would like you to answer this first question, and then you can kind of share it with each other and discuss it. So the first question is, hypothesize what could be causing many animals in a population to be affected by a similar type of cancer. Describe why and how it is spread, and try to come up with as many hypotheses as you can. And one thing we need to pause first and mention is that cancers are usually not contagious. If somebody has cancer, they're not going to spread it to somebody else. But what we're seeing here is that sometimes that can be different. So we're looking at how this maybe could spread and why it's so widespread in this population. And you already have some background and information about cancer. We spent chapter 5 in mitosis learning about cancer. So try to put some of that information to use here and think about how cancer forms, what it is, and how it could be spreading to like 50% of the population like we're seeing in these devils. So you can pause the video here, and I want you to write down 3 hypotheses on how you think this disease is spreading to these Tasmanian devils. So do that now. Okay, and I'm going to keep going. If you need to pause it longer, go ahead. So then if you could pick one of these hypotheses, I want you to choose one after you've shown a few people or shown the whole class, and then write down on your paddle, how would you test that hypothesis? So how would you design an experiment to determine if that hypothesis was correct? And just like we do when we design experiments, I want you to write your independent variable, which would be your treatment, your dependent variable, which would be what you're measuring, and your control. And then you can also write down some constants for that one hypothesis. So just choose one. Maybe you liked someone else's hypothesis that shared with you. One example might be maybe you think that this is caused from the food they eat. So your independent variable could be a specific diet. Your control might be like their normal diet that they had before. And then your dependent variable would be whether or not they had cancer. That dependent variable is probably going to be the same no matter what experiment you're designing. So that's just an example. And then the constants would be all the other things I kept the same. I would keep where they were held the same, like what cages they were kept in, how they were handled, other things they were exposed to, the water they drank, all of those other things the same. And the only thing I would change would be my independent variable, which would be the food, the diet, the new treatment diet, if I was trying to see if a certain diet was causing it. So let's pause the video here and you can write out your experiment. We just need those 4 things: independent variable, dependent variable, control, and constants. And if you're done, you can take a minute and share. You can pause this and share some of them and discuss if you think they're right. Remember that your independent variable would be the treatment. So you're trying to see what's causing it. And then your control would be the normal. Your dependent variable would be you're measuring whether or not they have cancer. So how many have cancer in the treatment group, your independent variable, how many have cancer in the control group, which is the normal group. Okay, so now we're going to move on to a study. Now, this is a real study that was done in 2006, and this is actually what initially piqued my interest in this. I stumbled on this study, and I thought it was really interesting. And then there's actually another study that came right after it that kind of followed it up, that I thought was really a great way to show how science can build on itself. And it also shows that when we do science, we don't just do like one little tiny field of science. We actually do, we connect a lot of different fields. So in this, we're looking at like genetics and cell biology, but we're also looking at like ecology, which is like a whole different field. But we have to know both of those well to be able to study this disease. So the researchers in this Pearson-Swift 2006 study, they became interested in this cancer and they wanted to look at the tumors. So they were looking at taking samples from the tumors and from a number of animals, and they were also taking samples of other areas of their bodies, like other cells that were healthy, and they were comparing those 2 cells together. And so here's a karyotype. Now, we looked at karyotypes before in the last couple chapters, and remember that these are the chromosomes that they're able to take during metaphase and kind of make a smear and then organize them to see what's going on. So this top A part, these darker chromosomes, these are chromosomes from healthy cells. So these would be like healthy tissue in the animal. And then the bottom part here is from the tumor. So they actually took samples from the tumor to compare it to the healthy cells. And they found that there are a lot of differences. So take a minute here. I want you to look at this picture of these 2 karyotypes and see if you can see some of those differences. And you can call them out, say them out loud if you notice some differences between the healthy karyotype and the cancerous one. What do you notice? Okay, I'm pretending I'm there and that I heard you. And I think I heard that this second chromosome is missing. Did you say that? and maybe this X and Y chromosome is missing because we have an X and a Y. Let me turn on my pointer. So we have X and Y here, it's missing. And then we're missing one of those 6 ones. And then we have all this whole like weird situation over here. These are like, we're not really sure where these belong. And some of these are also much shorter. So you can see that these are much longer, and you can see these are a lot shorter. So from this study on your paddles, you can erase the experimental design thing. I want you to answer this question. What do you suppose the researchers were trying to determine from their study? So we're kind of going like backwards. So we're looking at their results. What do you think the purpose of their hypothesis, or the purpose of their experiment? So really the hypothesis of what their experiment was. And you can write null or alternative. Remember, the null is that there's no difference between like a treatment and control. And the alternative hypothesis would be that there would be a difference between the treatment and control. So you can write your hypothesis, and then you can also write what you think their independent variable would be, their dependent variable, and their control. So I'll pause here for a minute. You can write down some of those ideas. And if you need to pause also and rewind and go back to that previous slide and pause it there, you can also look at that karyotype. I'll do that in a moment. So we're looking at this hypothesis, independent variable, dependent variable, and control for this previous study. So I'm going to rewind here and look at this previous slide. And what I would say in this study is that they were trying to determine if there was a difference between the healthy karyotypes, the chromosomes in the healthy cells, compared to the chromosomes in the cancerous cells. And what I would see here for their hypothesis, their null hypothesis would state that there would be no difference between the healthy cells and the cancerous cells. And then the alternative hypothesis would be that there would be a difference between the healthy cells and the cancerous cells. And then for the independent variable, I would use the cancerous cells, that would be like the treatment. The control would be the healthy cells, the normal ones that we're comparing them to. And then my dependent variable would be what I'm measuring. And I'm measuring how many chromosomes there are, or how long they are, or how they look. So that would be my dependent variable. So hopefully you got something like that, and you can pause and discuss if you maybe wrote something a little different. So from my interpretation of this and of the paper that I read, I found that they wanted to determine the origin of the cancer cells. They wanted to know where they came from and how they formed. And they actually came up with 2 hypotheses. So I pulled this directly from the table, or from the paper. Their first hypothesis was, did the mutations happen normally from the animal cells? So normally when we get cancer, it's our cells and they mutate for different reasons. Maybe it's a carcinogen, or maybe it's just a random mutation. It could also be something we inherit. And then those cells, as we know, they pass through those checkpoints when they're not supposed to, and then they're going through mitosis too much when they should be staying in interphase. And so that's how normal cancer would happen. We would just have mutations occurring for different reasons. Or their second hypothesis was, could it have been a cancerous cell line that was transmitted between animals, like an infection? So could it be that these cells were actually being transmitted from one animal to another and not actually coming from their own cells, which is normally how animals, how organisms get cancer. And we need to pause here and notice that we're not talking about an infection like a virus or a bacteria. We're not talking about that. We're talking about the cells, the cancerous cells of these animals actually being the infectious agent. So this isn't a virus. Don't use that word anywhere here. This is not caused by a virus, and it's not caused by a bacteria. It's just the cells themselves that would be transmitted from one organism to another, from one animal to another. So this is what they found out, and this is a quote from the paper. They said, we made the fortuitous observation of an inversion of chromosome 5. Inversion means that the chromosome was flipped in the karyotype of one animal. This anomaly was found in all cultures of that devil's normal tissues, but was not present in either of the chromosomes of his facial tumor cells. So we're finding that this particular animal had an inversion, a flip in one of the chromosomes, but the cancer cells didn't have that. The healthy cells had the flip, but the cancer cells did not in his facial tumors. So on your paddles, let's answer this question. Which hypothesis does this support? Does this support that this is just a normal cancer that's caused from our own cells mutating from this animal's own cells, or is it does it support the infectious cancer? And I want you to include evidence for your claim. If you think it's a normal evidence of normal cancer or evidence of infectious cancer, include that evidence. And I'll pause here for you to answer. You can take some time and discuss. Let's take a moment and see what different people around you answered for this one and what evidence they might have provided. You might have said that this is evidence of an infectious cancer because the cancer didn't have this inversion in chromosome 5 that all the other cells had in the organism's body. So that would be evidence that the cancer cells did not come from this, the organism's other cells. And so here's that picture again, and we would be looking here that there's, I'm not sure what that looks like, but there's an inversion somewhere on one of these chromosomes where a piece of it is just flipped, and they did not see that observation in this one. So they're finding that this karyotype did not come from the healthy animal cells at all, and it was not just mutated cells that turned into cancer. So this means that the healthy karyotype was from one healthy devil. The cancer karyotype actually came from 11 different animals, and they found that all of those animals, the tumors had exactly the same karyotype, which is really crazy. That imagine if you got samples from 11 different people who had tumors and all of their karyotypes looked the same from their tumors. That would be really unusual. So that was something really notable. So from that evidence, from the fact that all of these different animals' tumors had the same karyotypes, which hypothesis does that support? And let me get that thing away. See if it'll go away. Does this support that this is an infectious cancer or a normal cancer? And you might be able to answer this question pretty quickly. If you want to take a minute and write it on your paddles, and you can talk about why you think that. But you're hopefully you're discussing that this is evidence of an infectious cancer because all of these animals had they're different animals, different tumors, all of their karyotypes looked the same. So that's what we're seeing here from this evidence. And we also, what you guys probably noticed before, we're missing 2 of these chromosomes, and one of chromosome 6 is missing, and chromosome 5 has some anomaly. I think it's flipped right here or something. It looks lopsided. And then also the sex chromosomes are missing, and pieces are missing also. We said maybe there's some pieces that are messed up or missing. So they found all of these weird anomalies, but this was true of all of the 11 devils' different cancerous tumors. Even though they came from different animals, they all had these weird things going on, and they all looked like this, which is really unusual. So overall, this is what they found. Usually, if you took 2 people with cancer and looked at their DNA and looked at their chromosomes from their tumors or from their bodies, they would not be identical. That would be really weird because cancer cells are mutating independently in each individual person. But in this case, we found that the DNA from the tumors of these different animals was identical. So that's really unusual. That's our takeaway from this first study. So this is that first study. This is probably a lot of information. I want you to pause here, and you, in your notebooks, you can summarize what we found from this first study. So let's take a moment and do that. So hopefully you wrote that down. And now we're going to move into the next study that came after this, that followed up to this study. And it's different authors, Siddle et al. So Siddle and other people, more than 2 more. And this was the next year. And so they were actually going to specifically look at the DNA between healthy and cancerous cells. So they were looking at that. And they found that DNA from the cancer cells were very similar, like we saw in the last study. They were looking at that. And they found that if you look at the DNA from healthy cells, like from other parts of their body, not the tumor, those were very different, which is normal. You would notice that these animals' DNA sequences would be different. They also noticed that these animals were biting each other's faces when they were fighting, which we saw in the video. So based on just that part, I would like on your paddles for you to answer this question. Which hypothesis does this information support? Does it support that this is just a normal cancer, or that it's an infectious cancer? And you're probably able to answer this one pretty quickly too. This isn't a lot of new information. This is similar to what we saw in the last study, and we're going to dig into the study a little more. But we can see that this is probably supporting an infectious cancer, because we're seeing that the DNA from the cancer cells are similar, and from the healthy cells, they're not similar. And that's unusual. And then also we're seeing that these are aggressive animals that are biting each other. So I'm going to try to explain the procedures that they used to study the DNA. And this is called a DNA fingerprint. This might be something that you've seen on crime shows. And basically what we use it for is we try to match DNA. So in this example here on the right, we have DNA maybe from a crime scene, and then we have 3 suspects, and we can see that the bars match the suspect 2. And so that would be the person who was maybe the criminal. So hopefully you've seen something like that. And this technique is called gel electrophoresis. So what we do is we take pieces of DNA and we use these enzymes to cut them up into different places. But everybody's DNA is going to be cut up in different places because everybody's DNA is different. So even if you add the same enzymes to different people's DNA, it'll be chopped up differently. And then you put it in this little box. And this is a box that has sort of a jello in it, and you hook it up to electricity. And you have a negative electricity on this side and positive on this side. And then you put your little chunks of DNA right here in these little spots. You already cut them up. You squirt them in there, different lanes. These are like traffic lanes. And then you turn on this electrode, this current. DNA has a negative charge, so it's going to be pulled down to the positive. And you'll see these little bits here will be pulled down depending on how big they are. The little bits will migrate further, and the bigger bits won't migrate quite as far. And that's how we're able to tell if there's 2 strands of DNA that are identical, they'll get chopped up the same, and then the bars will be the same. So that's how we use gel electrophoresis. And this is a technology that they used in the second study. So the first thing they were looking at is cell surfaces. We know that cells have stuff sticking out of their surfaces, and they help like your body know if they're your cells, someone else's cells. This is like blood type. We have little A's and B's sugars sticking out of our blood cells if we're that blood type, and it helps our body know that like this is normal. So they were wondering if maybe the immune system of the Tasmanian devil was not detecting the little markers that were on the outside of cancer cells. When we have cancer cells, a lot of times they have little markers sticking out, and that's how our bodies know that they're cancer, and then our immune system will attack them. So that's a normal response that we do to cancer. But if we're not able to kind of keep up with that, that's one way that cancer can develop. And so they were wondering, why isn't the immune system just recognizing this cancer and killing it? And so what they wanted to see is, what if these surfaces of the cancer cells are the same as healthy cells? What if they don't have any markers on them? So what they hypothesized was, if the immune system is not able to recognize cancer cells, maybe the cancer cells don't have any markers on them. Maybe that's why they can't recognize them. So they did this technique, this gel electrophoresis that I just showed you, and they were looking at these markers on the cells. And so here in this picture, we have the T is a biopsy from the tumor. So those are tumor cells, those are cancer cells. And then we have healthy liver, healthy kidney, healthy spleen, and then M and N are kind of the control. So those are we're just making sure our gels are working. And what they're finding here is that all of them have the same bands. They all have the same markings, just like we saw here with suspect 2 and the crime scene. We saw matching bands, right? We saw a lot more bands in this case. We saw all these matching bands. And here, there's only one set, but they're all matching. So ignore the M's. Those are just the control. All of these are matching. What does that tell us? It tells us that the surfaces of the cells, of the cancer cells, looked exactly like the healthy cells. So these Tasmanian devils had no idea, their bodies, their immune systems had no idea that these were cancer cells. Their immune systems couldn't respond to them because the cell surfaces looked the same as the healthy cells. So let's answer this question on your paddles. Did they support or reject their hypothesis? They hypothesized, just as a reminder, that if the immune system isn't recognizing these cancer cells, that means the surfaces are the same. So was that supported or rejected from that gel electrophoresis study here? Does this support or reject that the cell surfaces were the same? So I'll give you a minute to think about that question. Does this support that the cell surfaces are the same? You can take a minute to discuss too with somebody next to you or the whole class. And what we do see here is that this indicates that the cell surfaces are the same because they all had the same band. So we're seeing that they all have the same DNA, and that the markings on the cell surfaces would be coded the same also. So that's why the immune system wasn't able to recognize and kill the cancer cells. So then they went on to another part of their experiment. They then wanted to, now that they know that the cancer cells don't have markers, that's a problem. They also wanted to see if these Tasmanian devils also had weakened immune systems. And they thought, what if their bodies, their immune systems are not able to recognize the cancer because they have a weakened immune system? So kind of a double cause here. The cell surfaces are the same, and maybe their immune system is weak. So they knew already from other research that marsupials are known to have a low immune system. So they studied the immune system of Tasmanian devils. They called them Con A devils. And then they studied regular devils. And I'm not sure how they enhanced the immune system of those devils. I don't know if it was like a food additive or an injection, I'm not sure. And then the third group was the eastern quoll, which is another marsupial, another type of animal, and that was the control group. So what they did was they took cells from another animal and exposed them to these animals to see how their immune systems would react. And this is what they found. So I'll try to explain this graph here. The black bars are the Con A, those are the immune enhanced devils that have like a better immune system. The MLR were the regular devils, and then the MLR eastern quolls were the control group, another type of animal, another type of marsupial. And they looked at over their immune response over 144 hours. They only measured the eastern qualls once. But as you can see here, the devils had a much lower immune response at all of the times compared to the enhanced devils, and then also compared to the eastern qualls. And all of these differences are significant. So they are finding, yes, they do have kind of a suppressed, weakened immune system. So how would we answer this one I just did? Was the weakened immune system hypothesis supported or rejected? So you can write this on your paddle. Would you say that this graph supported or rejected whether they had a weakened immune system? And so hopefully you said that that hypothesis was supported. We do see that they had a lower bars here, so they had a weakened immune system. So then these researchers went a step further, and they wanted to see if maybe this was because of low genetic diversity. So they looked at DNA sequences in the on the cell surfaces, and they also looked at DNA sequences for immune function, and they wanted to see if there was low diversity there. So they thought if the devils had lower diversity in those two sequences with their cell surfaces and their immune function. Okay, I'm recording right now. That was Lucas. So if they had a lower genetic diversity, they would be able to compare that to other species. So they looked at 3 groups. They looked at the devil, the Tasmanian devil. They looked at the Gur lion, which is a bottleneck species of lion. They're already known to be pretty inbred and low genetic diversity. And then they looked at humans. Humans have really big genetic diversity. We are very diverse genetically. So that would be kind of a control group. And here's the graph that they came up with. So the higher the bar, the more genetic diversity. And we're looking at the number of substitutions of different amino acids and how different they are. And then the lower the bar, the less genetic diversity. So we can see here these black or darker bars are humans, and then the gray bars are the Gur lion, and then the white bars are the devils. And you can see that the human bars are pretty high, so are the Gur lions. They're usually pretty high, and then the Tasmanian devils are down here at the bottom. So they're kind of consistently low. We're not seeing a lot of substitutions. That means we're not seeing a lot of differences. There's not a lot of genetic diversity. So does this graph support or reject the low genetic diversity hypothesis? And you can write that on your paddles and provide evidence, and you can share. And then we're going to look at the second and third hypotheses together. So do you think that together, and you can write these on your paddles, do these 2 experiments support or contradict each other? And so from hypothesis 2, from part 2, we found that they had a weakened immune system. And from part 3, we found that they have low genetic diversity. Do those seem to sort of agree with each other, or do they contradict each other? And you can talk about that too out loud and see what you think. And I know this is a lot of information, so I summed it up, I think on just one slide, so you can see it all together. All of these, so basically we saw 4 different studies and rolled up into 2. So here's what they found from all of this that we've kind of unpacked. They found that this disease does come from one cancerous cell lines, because the evidence for that is that all the cells are identical. And it started from one animal, and then it was transmitted to other animals. And then once they get like one cell kind of infected, then that cell will start to divide and turn into a tumor. But that one cell was from an original devil a long time ago, probably a couple decades ago. This is really unusual for cancers. And remember, we're not talking about a virus. This is not a virus. I don't want to see that on any of your papers. That's a common mistake, common misconception. This is not an infectious virus. It's not an infectious bacteria or other pathogen. This is cancer that is spreading from one animal to another. And it's one animal's cells that are starting to grow in another animal's cells because their immune system is weakened and they don't have cell surfaces to recognize that it's not supposed to be there. So this is really highly unusual type of cancer, and you wouldn't find it in humans. I don't know of any examples of this in humans. I do know of a contagious feline cancer in cats. There might be one in dogs that I think I've heard of from Mexico also. And you might have heard of others, but this is not common in humans, probably because one, we don't tend to have weakened immune systems, and two, we have a lot of genetic diversity. So we've got those two things going for us, right? So your take-home message here is not that cancer is contagious. It's that in this case with these organisms, we are seeing this example of a contagious cancer because of this really unique circumstance. And there are some viruses that can cause cancer, like the HPV, human papillomavirus, can cause cervical cancer, which I think we've talked about. So that is a type of cancer that can cause mutations in the cells of the cervix, and it would cause cancer. But otherwise, and that's not really a contagious cancer, that's just the virus that's contagious. Otherwise, cancers are not contagious. And the problem here was that the devil's immune systems did not recognize the other cells, the cancerous cells, as foreign because they had low genetic diversity and a weakened immune system. So those are your two reasons. And this might be a good place to pause and summarize. I wouldn't copy this, but maybe summarize it in your notes so that you have a good summary of what we found here. Okay, so now, and this might be Tuesdays. I don't know how long this video will take to get through, but this is what we're doing next. In your notebooks, you'll be proposing a solution. And so here's the background. Extinction is imminent for these animals, and they're in a pretty bad place. And so I want you to propose a solution that you think would prevent extinction for these species. And I want you to think back to everything you've learned about cells, how cells work, how they reproduce through mitosis, and how cancer can form, and also about genetics and maybe even ecology that we learned at the very beginning of the year and how to manage resources. And I want you to come up with a solution that you think would prevent extinction for these species. And so you'll have two paragraphs for this. The first paragraph will be explaining what happened. So that's paragraph 1. And then the second paragraph will be proposing a solution on how you can prevent extinction for these species. You can also get on Chromebooks and do a little research to see what's currently being done for these organisms. And please email me if you have any questions, and that should be the last slide.